Hello everyone, and welcome to our online lecture series for Bio 2. Today, we actually get into the tree of life, looking at the first three domains, which, if you remember from our systematics lecture, the domains of life are actually the highest order of classification. This is our earliest split. This is our most fundamentally different type of organism. It's essentially what that means. Being an early split means that these organisms have existed separate from one another. Their lineages have been separate from one another for the longest time period on planet Earth. That means they're going to be weird. Very different from what you and I are used to living as humans. These three domains of life are the bacteria, the archaebacteria, and the eukarya. Broadly speaking, the bacteria and the archaebacteria are our prokaryotes, meaning that they have no membrane-bound organelles. So they don't have things like a nucleus, they don't have things like Golgi bodies or endoplasmic reticulum. They are just organisms in one plasma membrane, DNA, ribosomes, machinery, all floating around in that singular membrane, bumping into each other anytime. This simple form of life arose in an aquatic, anoxic environment at some point in Earth's very, very distant past. We have, and we'll get into some of this evidence, but we have evidence of these first living things dating back to 3.5 billion years ago. This evidence of those first living things are actually fossils called stromatolites. And some of these stromatolites on the western coast of Australia are visualized here. A stromatolite is actually a sedimentary mat formed when minerals precipitate from water onto a prokaryotic microbial mat. So they're basically what happens when you get minerals forming alongside these microbial mats. And a microbial mat is exactly what it sounds like. It's basically just a large blanket of different little microbes, oftentimes are prokaryotes. Now, these stromatolites might not look like much, and you might question, how do we know that these are actually our fossils of microbes? Well, they're still forming today. On these microbial mats, which, if we look today, they're actually combinations of both bacteria and archaebacteria. forming on a wet surface. These microbial mats will actually still form stromatolites in our current environment. So we've looked at the fossil record that more recent things leave and found that there is similarity with some of these much, much older stromatolite fossils. So we started dating these stromatolite fossils and found that the oldest is 3.5 billion years old, but we've got some potential evidence that they can date all the way back to 3.8 billion years ago, which is quite old, considering that Earth, anyone know how old Earth is? Well, we estimate it to be about 4.6 billion years old. So in something like a billion years, we encountered, or life seems to have evolved on planet Earth. There's a lot of experimentation going into figuring out how exactly that life could have formed. It's not something that we'll spend a lot of time on in this class, but I do encourage you, again, to look up the Miller-Urey experiments and see what evidence continues to develop, even in the last few years, to better inform our understanding of how life could have evolved from this primordial soup. One of the major ingredients in that primordial soup, something actually very important to the formation of life, was that early Earth was not only partially aquatic, but it had no oxygen. It was anoxic. And we see evidence of that anoxic history of Earth in some of our rock formations. For example, in this sedimentary rock that's dated to, if I remember right, 3.15 billion years ago in its formation, you can see that some of the deeper layers, some of the first formed layers, are gray. This color of iron forms when there is not oxygen present, when it's not exposed or oxygen is not in the environment. We see oxygen in the environment turning iron red. What this rock therefore implies is that whenever it was forming, way back in the day, oxygen was not present. When oxygen was present in high abundance, and this was close to the surface, the iron that deposited was red. And then you get some, speckling of red, gray, kind of as this rock is either exposed or hidden from oxygen. Now, granted, any one given sample is not going to give us good evidence, but of course, we're not just relying on one rock to inform us that early Earth was anoxic. We look at all of the rocks that we can find that date back to 3-4 billion years ago and examine whether they contain iron or other substances and whether there is evidence of oxygen present in that environment. So these two bits of fossil evidence, one on the life side, seeing when organisms were alive, and one on the oxygen side, give us some insight as to what early Earth looked like. I'd also like to add on to that in addition to this primordial soup of early Earth that was aquatic and anoxic, we also have evidence of strong radiation. Strong evidence of radiation. Oops. The reason why radiation was much stronger in early Earth and would have really bombarded genomes in their early formation was that there was no ozone. Nothing protecting our planet from all of the damage that the sun can cause. We also get a lot of evidence of volcanic activity. Lots and lots of volcanic activity in early Earth. So that kind of sets the stage of what we're looking at in terms of early life. The fact that these first living things arose roughly 3.5 billion years ago, about a billion years into Earth's formation. That we have fossil evidence in the form of stromatolites, which stromatolites are sedimentary mats formed from microbial mats. And microbial mats are just combinations of archaebacteria and bacteria that live on a wet surface that still form today. These early life forms, these prokaryotes, have very specific roles in our environment today. They have a vast ecological niche. They occupy so many different spaces. They are unicellular, but they do have some tendency to form multicellular amalgamations, meaning combinations with other things. They oftentimes are responsible for recycling nutrients. They oftentimes are kind of the things that put our different end products back into the system, so that way products can be reused, so they are part of the recycling component. And we know that these prokaryotes lived in this anoxic environment for roughly 2 billion years. Before these prokaryotes were responsible for changing our environment in what is called the oxygen revolution. They were responsible for converting our planet from a low to no oxygen environment to a high oxygen environment. They made our ozone. And by they, I specifically mean the lineage of bacteria that are currently around and able to evolve or produce oxygen. Those are our cyanobacteria. You might better know them as blue-green algae. We call them algae because they are aquatic. Again, aquatic environment, right? And they are have the color of being bluish green. But unlike all other algae, they are not eukaryotic. They have no nucleus. Instead, these are incredibly small organisms that still to this day form a massive component of our aquatic ecosystems in that they produce sugar and oxygen. So they are still to this day major producers of oxygen on our planet. A lot of these stromatolite fossils are thought to actually be large microbial mats of cyanobacteria. Additionally, cyanobacteria are thought to be the endosymbionts that are today chloroplasts. They evolved the ability to perform photosynthesis with all of the enzymes like rubisco. And rather than reinventing the wheel, the lineage that became algae and plants, the eukaryotic lineage that did so, engulfed one of these cyanobacteria and still uses a lot of those same, or exactly those same machinery as cyanobacteria to perform photosynthesis. Cyanobacteria have enzymes like rubisco and produce pyruvate in their Calvin cycle, in their different steps of photosynthesis. And we see the exact same thing occurring in our algal lineages. But more on that when we get to it in our next lecture on eukaryotes. So, what to make of this first introduction? Well, keep in mind that for now, Earth is an aquatic anoxic environment, having these very simple prokaryotic organisms. Life spawned from this primordial soup, and we started to see massive changes in the environment. Again, these changes in our prokaryotes occur over immense timescales, much, much slower, seemingly, than a lot of the changes that occur much more recently in our multicellular life forms. So in terms of timescale, this first lecture on prokaryotes is going to cover much more time, 3 to 4 times as much time as the rest of our lectures on biodiversity in this class. It took life, seemingly, quite a very long time to evolve the complexity of multicellular life forms. That does not mean that these early life forms are, quote unquote, simple in their metabolisms. Their vast ecological niches have allowed them to diversify immensely. So rather than having one complex life form that occupies a broad ecological niche as a single organism, you have millions, potentially billions of different species of bacteria that are specialized on different ecological niches. Now, in terms of their relationship, this 3-domain system of life is what we have as our best hypothesis to this day, though I will inform you that this may change in the future. There is growing evidence showing that perhaps eukaryotes are just one of the branches of archaebacteria rather than being sister and completely separate to archaebacteria. We'll talk a little bit about that when we get to the archaebacteria in a bit. So, that first leads us to that incredibly diverse group of prokaryotes called bacteria. So, bacteria are obviously incredibly diverse. We oftentimes classify them based on their physical structure. Those 3 structures are seen above here. They are the spiral bacteria that are known for their spiral shape, no fancy name there. The bacilli bacteria, these are the rod-shaped bacteria. So here's our spiral. Here's our rod. I will also have you note, look at the size difference between the spiral and the rod. The spiral are generally a little bit larger, depending on the species. And then our cosi, the circular bacteria. In terms of their structure, what cellular features do all of these bacteria have that unify them as bacteria? They are all made up of some sort of cell envelope. In many cases, a cell wall, as well as a cell membrane. More on both the cell wall and cell membrane later when we look at some comparisons between them. They have a cytoplasm, a liquid on the inside that is holding both their nucleic acids and their ribosomes, abilities to make proteins. Taking up too much space. They typically can have an appendage on the outside. That appendage oftentimes helps them relocate and is a flagella. The boot print of the flagella, the fact that this organ is so ancestral to our tree of life is carried within us today, as there still are human cells that carry flagella very similar to the flagella that we see in bacteria. Those human cells are obviously sperm. So if we're going to go ahead and put a tick mark on the tree of life, we can see that this universal ancestor seems to have had a flagella, and that flagella is inherited both by the domain bacteria and the domain archaea and the domain eukarya. Now, bacteria also have a wide variety of energy sources in filling so many different ecological niches. They are both phototrophs, like our cyanobacteria, as well as chemotrophs, breaking down chemicals, oftentimes sulfur-based chemicals, for their energy. They also have very unique forms of reproduction that will seem familiar to you based on our discussions of meiosis. Their reproduction is done via binary fission, which is basically a cell splitting in two, kind of like mitosis. They can have bacterial sex in two different ways. They can perform transformation, which is the uptake of a plasmid from the environment, meaning that if there's a piece of circular DNA floating around them, that they can absorb it and then all of a sudden start expressing the genes on it. We've used that to our great benefit in genetic modifications and the production of life-saving medicines like insulin. We stuck an insulin gene on a plasmid into E. coli and have the E. coli make our insulin. So that way, diabetics are not doomed to shorter, more painful lives. Thank you, bacteria. And then they can actually have sex in the wild through the process of transduction, wherein a bacteria will form a structure called a pilus and insert its genome into another bacteria, thereby allowing for some of the benefits of reproduction within these unicellular prokaryotes. So quite different from us, but we see a lot of parallels in their strategies. Now, as far as the diversity of these bacteria go, we can take a look at the phylogenetic tree here to look at the next level of organization. From domain, we drop down to do king's play, so kingdoms. For whatever reason within the bacterial lineage, we don't really divide them into kingdoms. So these groups here are actually phyla, or phylum. Phylum from plural, phylum for singular, phyla for plural. So these are the different phyla that we group our domain bacteria into. The first and incredibly diverse are our proteobacteria. The proteobacteria are named because they're actually named after the Greek god Proteus, a god of sea and rivers, who is capable of assuming many different forms. So just like water takes, lakes, rivers, streams, etc., these bacteria are named thusly because they actually also have lots and lots of different forms. Within the phylum Proteobacteria, we have the different classes of alpha proteobacteria, beta proteobacteria, gamma proteobacteria, delta proteobacteria, epsilon, zeta, acido, hydreno, oligoflexia. It's just incredible how much diversity we have within this group and all of the different life forms that they take. I will not expect you to know the different alpha, beta, gamma proteobacteria. Just know that that this group is incredibly diverse. They are a gram-negative group of bacteria, so we'll talk about that, but that means that their cell wall does not have the structure that would give it a purple stain with the gram stain. So that means that they have a thin peptidoglycan cell wall. We'll actually take a look at the cell walls in a few minutes here after we go over a broad survey of diversity. The proteobacteria have a huge number of pathogenic genera, including things like Salmonella, Yersinia, which is our bubonic plague. And if there's one thing that you'll find out in taking any of my classes, I will take any opportunity to talk about the bubonic plague. Very interesting species, very interesting role in human history. Yersinia pestis is actually the bubos, and it's one of our gamma proteobacteria. We've also got in here, within the gamma proteobacteria, a good gamma proteobacteria, Escherichia coli. And no, I will not write out or even attempt to spell Escherichia. That's why everyone calls it E. coli. This is one of our good gammas. But of course, there are some toxic form of E. coli that are not good for our digestive tract. But generally speaking, there are good E. coli in our digestive tract. We also find within the proteobacteria, different nitrosomes, which are bacteria that are able to fix nitrogen. These are within the beta alpha proteobacteria. And then we also have Heliobacter pylori. In the epsilon group, that E-looking thing for epsilon, that is a digestive disease in humans. So you may have heard of Heliobacter pylori because they actually form ulcers in humans. So it's an incredibly wide-ranging group that has a gram-negative stain because of their thin peptidoglycan cell wall. We also believe that it is that they have we do not believe, but rather we know that this group has the nitrogen fixation. So let's go ahead and write that out with a star, since that is a very, very important function. And since we noted the source of our chloroplasts in plants, I also want to note what is the current thinking on what is the source of the mitochondria, and that is our proteobacteria. We think that a lineage of the alpha proteobacteria actually is the endosymbiont for mitochondria in eukaryotes. So clearly a very diverse, very cool, but also pathogenic, or at least cool biologically, group of organisms within the within all of the proteobacteria. Next up, we have the phylum of Chlamydius. Or the Chlamydiae. This is another group that's remarkably diverse, both pathogens and symbionts. So I guess, not sure if I defined this term yet, but pathogen means that something that's pathogenic means that they are able to get organisms sick or have negative negative effects. They produce disease in organisms. Whereas a symbiont is an organism that also lives inside of another, but is beneficial. So they are quite polar opposites of one another. Pathogens live inside of something and make it more difficult for that something. Symbionts or endosymbionts live in something, but then cause it to do better. The chlamydia are, again, remarkably diverse, both symbionts and pathogens in different lineages. We haven't studied their marine forms, but we know that they do exist. They seem to be very good at infecting eukaryotic hosts, but we can't actually study them very well because they don't propagate in a lab very well. We have to study them inside of their hosts. The members of chlamydia cause diseases like chlamydia pneumoniae, which is a very specific type of pneumonia caused by a member of this group, as well as the disease chlamydia itself, the sexually transmitted infection, chlamydia. Chlamydia is, of course, the most commonly transmitted bacterial STI in the US, with something just under 3 million chlamydia cases a year. That is a yikes. Next up, we've got spirochetes. Which, true to their name, are the spiral form of bacteria, given that shape. They have that shape because of the fact that their flagella, which acts more like an axial filament, runs lengthwise on the inner membrane and outer membrane, because they have two membranes. Which in some groups of phylogeneticists make them diderms. So we probably won't get into that, but should you get into bacteriology, you may need to be familiar with the term diderm or double membraned. And that gives them kind of the weird spinny movement pattern that's characteristic of spirochetes. Now, there are two different groups of spirochetes that you can be familiar with because of their pathogenic nature. Lyme's disease, the tick-borne illness, is a spirochetes, and syphilis, the STI, is also a spirochetes bacteria. We've already talked about our cyanobacteria, these being our most abundant group. And actually, I will re-emphasize how important that is. So spirochetes get our most abundant lineage here, and we're going to be doing this for a lot of our different lineages, talking about where the most phylogenetic diversity is maintained in different lineages, just to give us an idea about which strategies have been more or less successful. And of course, a lineage that is able to feed itself based on light and occupies aquatic environments, how much of the earth is surrounded in water, is going to be an incredibly abundant and diverse group of bacteria. So certainly the most bacteria on planet Earth are of this cyanobacteria type. Our last group are our gram-positive bacteria, which after this discussion, we're actually going to go into what it means to be gram-positive versus gram-negative. The gram-positive bacteria are comprised of bacteria like anthrax. It's not just a poison, it's a toxin created by a bacteria. Botulism, the bacteria that can inhabit canned foods and cause cans to swell up. So please never eat a can that is swollen. It has botulism. And you may be familiar with botulism in the minor plastic surgery of Botox because it is the botulism toxin that is injected into oftentimes facial muscles to cause them to be paralyzed. If they are paralyzed, they do not wrinkle. So yikes. Botox is the practice of inserting a muscular toxin to paralyze and relax the muscles so that way they do not wrinkle. Interesting choices. And then we have Clostridium difficile in the gram-positive group. This is the one that oftentimes whenever you take antibiotics and you get diarrhea afterwards, it is because the antibiotics have wiped out your beneficial microbes in your gut and have made room for Clostridium difficile. And Clostridium difficile makes it difficult to maintain a proper digestive system. So they cause you to have diarrhea. So they are this antibiotic opportunist. Your gut will return to good health when the good microbes have beaten out Clostridium for that top spot and have regained their function. So now let's go ahead and take a look at what exactly our gram-positive versus gram-negative bacteria have and don't have. Gram-negative bacteria are frequently having this, or always having this outer and inner membrane, so two membranes with a periplasmic space that contains a peptidoglycan cell wall. Peptidoglycan is a polymer that is made of both derivatives of peptides or proteins and sugars, glycans, to provide structure and strength to the bacteria. The cell wall itself of bacteria is there to prevent osmotic lysis. Which, depending on how good your biology vocabulary is at this point, means water popping. So you can imagine that an organism that has no limit as to how much water it can imbibe would just continue to take in more water. The cell wall prevents those bacteria that are living in fresh water or highly aquatic environments from popping because they put an outer limit as to how much water can be taken in. Plants also have a cell wall, largely for very similar purposes. Animal cells do not have a cell wall, and when they are put, like when your red blood cells are put into pure distilled water, they will actually explode because too much water will rush in and they will pop. The gram-positive bacteria and some proteobacteria have these thicker peptidoglycan cell walls, whereas the gram-negative bacteria are all of the other different types that we have discussed thus far. So that is our quick introduction into the diversity of the domain bacteria and several of their phyla. I won't ask you to really keep track of the taxonomic designations of phyla versus kingdom, but just do know that we're talking about things within the domain bacteria. Next up, we are going to take a look at our archaea. Now, it is quite useful to take a look at our archaea in terms of their similarities and differences with the bacteria. So to highlight some of these, and this is table 22.2 in your textbook, both bacteria and archaea are prokaryotic. Both of them have highly variable cell morphology. The bacteria have peptidoglycan in their cell wall, whereas the archaea do not. So this is one of those very unique differentiating features. Archaea do not have peptidoglycan. Another unique thing is that the cell membrane of bacteria is always a lipid bilayer, where you've got a phospholipid top, phospholipid bottom. So that is a bilayer, not the fact that there are an inner and an outer membrane. You've got this phospholipid bilayer present in each of the plasma membranes, whereas some of the heat-adapted archaebacteria have a lipid monolayer. And we'll talk about why that is when we take a look at that diagram. Their chromosomes, as we've discussed before, typically circular for both lineages, whereas eukaryotes have our typically linear chromosomes. Another unique feature of bacteria is that they have a singular place of DNA replication. So they have one starting point, whereas in archaea, they have multiple, and this is in common with eukaryotes. So if you were potentially graphing out another tick mark that's unique to eukarya and archaea, you could put multiple origins of replication. Just trying to, obviously this is kind of a niche one, but just trying to rekindle this idea that you should be in your notes drawing out tick marks on the tree to keep track of what changes happen when and what changes are unifying or apomorphies to different groups. The vocabulary gets away from you pretty quick if you don't stay on top of it, so please make sure to stay on top of your vocabulary. Hopefully apomorphy has meaning to you. Other similarities are more molecular, looking at RNA polymerases, whereas bacteria have singular, archaea have multiples, just like eukaryotes do. We have whether the Calvin cycle is present in terms of photosynthesis. Archaebacteria are unable to do that, whereas bacteria are. So let's again take a look at the diversity that we have in archaea, just like we did in bacteria. There are several different groups, and I will struggle to pronounce these. I'm not even sure I'm pronouncing them correctly, but you have the euryarchaeotes, the crenarchaeotes, the nanoarchaeotes, and the core archaeotes. Might be archaeotes. Honestly, never heard another human being pronounce them. Have always just read them. They each have different ecological roles. That's kind of why we group them as such. The euryarchaeotes are methanogens. So you can think of the euryarchaeotes, or thank the euryarchaeotes for the methane in our farts. And as an educator, I will always take any opportunity to write that word or say that word out loud. So, yep, that's that. The crenarchaeotes are oftentimes found in volcanic springs. Hot, acidic, and sulfurous environment. The nanoarchaeotes, we've only found one species. And it's likely not because there's only one species on the planet, but because they are very, very difficult to identify and find. They've been found on the bottom of the ocean as well as in Yellowstone. So clearly they occupy quite a wide and old variety of niches, but they're just incredibly difficult to find. And then the core archaeotes, we've never been able to culture. And I mention that because the common thread of domain archaea is that they live in places where nothing else wants to. They are extremophiles. And some different forms of these extremophiles are found in this table here. Like acidophiles, they live in acidic environments, like pH 3 or below. For a point of reference, lemon juice has a pH of something like 2. That means that these organisms live in the equivalent of lemon juice. Now, I don't know if you've ever experienced the absolute joy that is lemon juice getting into your eye, but lemon juice is not very pleasant an environment to live in, much less even come in contact with to your open membrane surfaces. We also have extremophiles that live within pH above 9, alkalophiles, or thermophiles that like it hot, or hyperthermophiles that like it ridiculously hot, so live in over boiling water. Psychrophiles, which live in extremely cold environments, so even below freezing. Halophiles that live in extreme salt environments, and osmophiles that live in extremely high sugar concentration, which is absolutely bizarre to me, but you'll note that sugar can be very, very difficult to live in. You keep honey out on your countertop, and it does not spoil for years and years and years because that amount of sugar is very difficult to culture organisms within. So most of our emphasis on our archaebacteria is going to be this discussion of the fact that they are extremophiles. The other 2 points about archaebacteria that I want you to note is that lipid monolayer in some cases, and then the presence of eukaryotic signature proteins, as these make for excellent tick marks on our branch that unifies the domain archaea and the domain eukarya. So the story behind eukaryotic signature proteins is that when we first started looking at these different organisms, we started to identify proteins that we found specifically within eukaryotes that differentiated them from archaebacteria and bacteria. So we figured that eukaryotes were their own branch. They were too different from everything else. But we started studying archaea more and more. And as recent as 2015, when a larger survey of these things were published, studies of archaea are kind of far and few in between because they live in such difficult environments. We have found an abundance of different proteins that were classified as eukaryotic signature proteins, so again, those specific proteins to eukaryotes in our archaebacteria. This included relatives of actin, a cytoskeleton protein, and tubulin, another cytoskeleton protein that we thought were specific to eukaryotes. On top of that, a lot of proteins involved in, or several proteins involved in transcription and translation. Are also shared with archaebacteria. So the more that we study the molecular phenotypes of archaebacteria, the more evidence we found for this relationship. And this also leads us to perhaps what the future of phylogenetics, as we continue to uncover relationships within archaea, with the idea that eukaryotes are actually potentially wedged within the archaebacteria. They are just a subset of the archaebacteria, a branch off of this group. Now that is not the current accepted hypothesis, but it is a hypothesis out there. So it'll be curious, and it does have a fair bit of support for that hypothesis, again, largely in that 2015 publication. But we'll see over the course of my career as to whether this classification changes. You'd figure that something so basal as the 3 major domains of life would be something that science has resolved fairly well, but science always responds to data. So we'll see how all that changes. The last little bit that I want to mention here has to do with the phospholipids. So when we're looking at the bilayers of phospholipids, so again, these are the structures, the molecular structures that make up the membranes. They have a hydrophilic, so a water-loving outside, and a hydrophobic inside. So that means that water can exist on one side and the other, but never in the middle, never at those hydrophobic tails. However, in environments where there is a large amount of heat, the fact that there is a break here can actually cause that phospholipid layer to break up. In order to cope with the extreme heat that some of those thermophiles live in, the phospholipid bilayer that is present in archaea actually fuses all the way across, providing more resilience to heat. And so I include this because this is one of these molecular adaptations that we don't appreciate because we can't see them with our eyes. But once we start looking at the exact structure of these chemicals, we start to really see how these small changes molecularly can allow organisms to live in environments that we would not even dare stick our hand into for a split second. Boiling water is not something that you want your cells in for any length of time. Yet these organisms that share ancestry with you can live there their entire lives. Isn't biology incredible? Or maybe I think so because I'm a biology professor, right? So that's where we'll end it today, looking at the diversity of archaea and some of those tick marks that are shared. So please make sure that you are adding that information onto your tree of life. By the end of this whole section, you will have an absolutely ginormous tree of life that hopefully is fleshed out with a whole bunch of different tick marks to highlight the different evolutionary transitions that occurred over time. Try to keep track of some of the massive diversity that we get in bacteria, particularly within the shapes for our vocabulary quizzes and some of their other features. And then I will see y'all next time for our discussion of our eukaryotes.